Hi everyone, welcome to our webinar today. This is for researchers and developers from non-clinical to clinical level endpoints, MASH HCC drug development using the STAM model, therapeutic evaluation of resmetirom and semaglutide in the STAM mouse model, bridging surrogate and clinical outcome. Just a little bit background and objectives of this webinar. So recently, the resmetirom and semaglutide have been approved by the FDA for MASH patients. However, they are only based on histological surrogate endpoints such as NAFLD activity score and fibrosis, demonstrating efficacy for clinical outcomes such as HCC liver cancer, also liver failure, transplant complications, or survival still remain a major challenge. STAM mice model reproduces the full spectrum of disease progression from MASH to fibrosis and HCC and death within 20 weeks, which enables the evaluation of not only surrogate indications, but also the clinical outcomes. The objectives is to evaluate the effect of resmetirom and semaglutide in STAM mice during the fibrotic phase, to assess improvement of histological surrogate endpoints such as NAFLD activity score and fibrosis, to establish the foundation for future assessments of clinical outcomes such as HCC incidence and survival rate. This webinar provides practical insights for researchers and drug developers on bridging surrogate endpoints to clinical outcomes. This is an overview of STAM mice model. We have 6 key features. The first one is a disease progression mimics human MASH. STAM replicates the full disease progression observed in humans, from steatosis through MASH to fibrosis and hepatocellular carcinoma, enabling high relevant study outcomes. Secondly, this model have a rapid spontaneous HCC development. Unlike other models, which takes 24 to 68 weeks to develop HCC, STAM model achieves spontaneous HCC by 16 weeks, offering a much faster preclinical timeline. The third feature is a comprehensive endpoint assessment. This model allows evaluation of both surrogate markers such as NAFLD activity score and fibrosis, and critical clinical outcomes, including carcinogenesis and survival, providing a holistic model readouts. The number 4 is the strong patent protection. The STAM model is patented across Japan, the US, EU, and Korea, ensuring exclusive rights and trust in its proprietary technology. The 5th is proven track record. Over 1,500 compounds have been evaluated using STAM model, including the newly launched STAM HCC IO Plus model with 100 compounds tested in 2024, demonstrating robust validation. Number 6 is unique advantages over other models. STAM model offers the integrated fibrosis, HCC, and survival data in a single model, outperforming competitors that only deliver surrogate endpoints, enhancing translational research value. The next is a comparison of mouse model. Here are several mice model exist for MASH indication, but most replicate only partial features and do not progress to advanced stages. STAM model uniquely reproduces the full spectrum from MASH to fibrosis, HCC, and death within 20 weeks. This enables evaluation of both surrogate and clinical outcomes. As you can see, STAM model meets most of characteristics which are prone to MASH patients. We can evaluate efficacy for NAFLD activity score improvement, fibrosis, resolution. Those are surrogate endpoints. Also, clinical endpoints such as HCC. It develops tumors in very short period of time compared to other models. We also see evaluation of ammonia in blood and MELD score. It's short for model for end-stage liver disease score, such as decrease in albumin, increase in total bilirubin, increase in prothrombin. STAM is so far the only mice model that can evaluate mortality, which is one of the key indications for clinical outcome. At SMC Laboratories, we offer a wide range of mouse model, and we can accommodate any models listed here as well. GAN diet, CDA-HIFA diet model, Western diet and CCL4 model, MS MASH model, high-fat, high fructose model, or LDA-L receptor knockout mice. In preclinical phase, our clients often run multiple mice models to further understand their efficacy and gain more confidence before moving to clinical phase. Comparison of mice models, comparative analysis of MASH therapy models. The STAM develops spontaneous HCC within 16 weeks, much faster than GAN diet model, which is 68 weeks, or CDA-HIFA diet model, which takes 60 weeks. Secondly, it enables both surrogate endpoint assessments and clinical outcomes. Other models show partial surrogate endpoints but lack comprehensive outcome data like survival or transplantation. And lastly, the STAM model uniquely integrates fibrosis, HCC, and survival readouts, providing early access to clinical outcomes. STAM is the most comprehensive and translational MASH model, bridging preclinical data to clinical outcomes. Next, we will talk about the methods of STAM model creation. STAM mice model is established through a two-step process that combines chemical induction and dietary intervention. Newborn mice receive a low dose of streptozotocin to induce mild pancreatic beta cell injury. After weaning, the mice are fed a high-fat diet to accelerate the progression of liver disease. As a result, the mice sequentially develop steatohepatitis, fibrosis, nodular lesions, hepatocellular carcinoma, and ultimately death within 16 to 20 weeks. In this study, treatment with resmetirom or semaglutide was initiated at week 9 during the fibrotic phase, and animals were sacrificed at week 12 for histological evaluation. This is an example of study design. STAM mice were randomized into 3 groups, which is vehicle, resmetirom, and semaglutide. Treatment was initiated at week 9 during the fibrotic phase and continued for 3 weeks until week 12. Histological and biochemical analysis, including NAFLD activity score, fibrosis area, ALT, and AST were performed to evaluate drug efficacy. Here was the results of biochemical markers. Biochemical analysis demonstrated that semaglutide significantly reduced plasma ALT and AST levels compared with the vehicle group. This indicates an improvement of liver injury. In contrast, resmetirom did not show a clear effect on these biochemical markers during the treatment period. The next result is for the histology images. Histological evaluation was performed using H&E and serous red staining. H&E stained sections were assessed for steatosis, inflammation, and hepatocyte ballooning. Now, for the activity score was calculated according to established criteria. Serous red stained sections were used to quantify fibrosis area. Images were captured at 200 magnification in 5 random fields around the central vein, and the mean value was calculated at the fibrosis area for each animal. Here was a result of NAFLD activity score and fibrosis. Both resmetirom and semaglutide significantly improved the NAFLD activity score and fibrosis compared with the vehicle group. These results confirms that both agents exert histological benefits in the STAM mice model. When NAFLD activity score was analyzed by subscores, semaglutide markedly improved the steatosis and lobular inflammation, while resmetirom showed a strong effect on hepatocyte ballooning. These results indicate that the two agents exert distinct patterns of histological improvement in STAM mice, reflecting their different pharmacological profiles. Both resmetirom and semaglutide significantly improved the NAFLD activity score and fibrosis in STAM mice model. These distinct profiles are consistent with findings from human clinical trials. STAM model uniquely reproduces the full disease spectrum from MASH to HCC and death within 20 weeks. This model bridges surrogate endpoints with clinically meaningful outcomes, supporting its value as a translational preclinical platform. The conclusion here is the STAM model provides a valuable translational preclinical platform that supports meaningful assessments of both surrogate histological endpoints and clinical outcomes. How was today's webinar? This is the end for today's discussion. You can visit our LinkedIn page. We're planning on the hosting another webinar, and we would like to hear back from you. Until next time, we'll see you soon. Have a good day.